Okay, welcome back everybody. Today begins the sort of first day in a several chapter series of exploration of biomolecular structure and function. Yeah, this is very exciting. Now, there are some good things and some bad things about this section of the class. The bad thing is the molecules we'll be dealing with are large enough that you're not going to be able to draw them very quickly. So we're going to be relying on your ability to focus in on the most important aspects of a molecule and focus on that and ignore the other stuff. These are skills that you develop by thinking about functional groups, and that we're going to continue to need you to do that. But the key and the other disadvantage is there's so much detail that if your main strategy in the course is content delivery and information retrieval, you're going to run out of RAM. You're going to want to know at every point, do I need to memorize that? Do I need to memorize that? The answer is usually no, unless I say otherwise. But what you want to focus on is the relationship between structure and function. That's sort of the key principle to take from this section of the course. The structure of a biomolecule determines its function in predictable ways that you can learn about and master. So I'm going to warn you today, because I don't want to, if we spend time drawing every single thing together, we're going to run out of time to describe the important things. I'm going to use some drawings that I have prepared previously. You will hate that. It is okay. You can hate that and you can write to me about it. I would be happy to read those emails. Delete. No, I don't delete them. No, I'm just kidding. But the slides and the lecture notes will be available to you. You can look at them. And in many cases, they're going to reproduce some things that you've seen already in the textbook, but with a little bit added detail. All right. The first thing we need to talk about in this chapter 22, which is on amino acids and peptides and proteins, is the difference between an amino acid and a peptide and a protein. So an amino acid is, the ones that we're going to be learning about are called alpha amino acids. They are called alpha amino acids because the amino group, or the ammonium group, is on the alpha carbon relative to the carboxylic acid. Now, you'll notice I've drawn it this way to draw your attention to the fact that an amino acid has a couple of different groups on it, a couple of different acidic protons. You've got really the carboxylic acid group, which, as you've learned from your text, has a pKa around 2 for an alpha amino acid. The reason it's lower than expected for a carboxylic acid, usually you expect around 4, is the impact of this electron withdrawing nitrogen, this electronegative nitrogen on the alpha carbon, which through an inductive effect pulls electron density away. Now some of you are like, hold on, nitrogens with lone pairs are supposed to be electron donating groups. Yes, when they're conjugated to benzene rings or other things. This nitrogen is not conjugated to that one, and by the way, it's no longer got a lone pair because it's protonated. Okay, so that's why that pKa is lower. Similarly, this ammonium group on the other side of the amino acid has a pKa a little bit lower than expected for a typical ammonium ion. Normally they're around 11. This one's 9, and again, the reason for that is the nearby electronegative oxygens that are withdrawing electron density via an inductive effect. I'm just going to write that over here to help you remember why those pKa's are a little bit lower. Now, most of the alpha amino acids have a side chain. We will talk about that in a few minutes. Differences, all the alpha amino acids have the same basic structure. Carbonyl carbon, alpha carbon, nitrogen, we call that the backbone. And then over here, we call this the side chain. I'm going to use those terms liberally without even noticing it. So if you don't know what I mean when I say backbone versus side chain, you better figure it out. And that's why I am demonstrating it now. Backbone, side chain. Got it? Now that's just one amino acid. Your nature uses an alphabet of 20 that are encoded for in ribosomal protein synthesis. There are others that are used and get incorporated, but we'll focus on the 20 what are called standard or canonical amino acids, and we'll learn what their side chain structures are. But before we do that, I need you to know the difference between an amino acid and a peptide. So I'm going to draw a simple dipeptide. A dipeptide is where I have two amino acids that are linked together through an amide bond. So this is side chain 1 and this is side chain 2. Notice, by the way, that those alpha carbons are stereocenters, and that will be true for almost all of the alpha amino acids. They all have this configuration, where if you draw the alpha carbon pointing down with the carboxylic acid on the right and the amine on the left, the side chain is a dash instead of a wedge. They all have that configuration. Why am I not saying they're all R or they're all S? Because depending on what the side chain is, it changes whether they're R or S. But they all have this configuration. And in fact, sometimes biochemists like to use an old archaic way of talking about stereochemistry. They will use the letter L. This is an L amino acid, and that's fine. We can. It just means they have that particular configuration. Now, this is a dipeptide. You'll notice that we have an amino acid, a side chain here and a side chain there. The side chains could be different. Here a side chain, there a side chain. I know, we're all old McDonalding. Sorry about that. Not as much on the brain as it used to be. It's been probably 15 years since I've sat down. No, 12 years since I've sat down with a child at the piano and sung Old MacDonald. Sad. Oh, nostalgia, right? Whatever. This is a dipeptide, and we have an amide bond that links the amino group of one amino acid with the carboxylic acid group of the other. Interchangeable and synonymous with amide is peptide bond, okay? But it's important that you be able to look at a dipeptide and conceptually break it into two parts. You have amino acid 1, and then you have amino acid 2. This nitrogen belongs to this amino acid. This carbonyl carbon belongs to amino acid 1. And we will call different amino acids within a peptide. We will sometimes use the term residue to describe an individual amino acid within a larger peptide. So somebody will point at a sequence and say, this residue right here, and you're thinking, residue of what? No, it's not used in that sense. We're just using the term residue to refer to an amino acid that's within a protein. Let me, or a peptide, let me draw a dipeptide for you, a tripeptide for you, and then we'll talk about how the peptide or protein structure has changed by introducing additional amino acids. Pay attention to the fact that above there was a side chain on one end. Amino acid 2 was on this end and had the free carboxylic acid. Amino acid 1 was on this end and had the free ammonium group. That's going to change when we go from 2 to 3. So this is a tripeptide. I promise you I'm not going to draw a tetrapeptide. If you break this up into its component parts, there's one amide bond or peptide bond. That is the bond between amino acid 1 and amino acid 2. Here is the next peptide bond. And that breaks the molecule up between amino acid 2 and amino acid 3. Now, notice the difference in environments. Amino acid 1 is lost its side chain. Sorry, I guess I was in a hurry. Notice that amino acid 1, on its alpha carbon, it has this charged ammonium group. Notice that amino acid 2 doesn't have a charge group on it, right? The alpha carbon is not adjacent to a charged ammonium group. Notice that amino acid 3 its alpha carbon is adjacent to a carboxylic acid. Now let's go ahead and draw this molecule as it would appear at physiological pH. Quiz time, pKa2. Should that carboxylic acid have its proton at pH 7? No, that thing's gone. You better be able to do that. So we should erase that proton and put a negative charge in its place. Do you see the difference in environments? Amino acid 3 has a negative charge on one side. Amino acid 1 has a positive charge on the other side. And the amino acid in the middle doesn't have any charges around it. Those are distinct environments. We have ways of talking about these unique environments in peptides and proteins. We call the end of a peptide or a protein that has the amino group on it, we call that the N-terminus. Okay, or that's short for the amino terminus. And then the end of the peptide that has the carboxylate group at it, we call that the carboxy terminus, or for short, almost everyone will say the C-terminus. N-terminus, C-terminus, different environments. You have to know about that. It's obvious if you know about the charges. A positive charge is different from a negative charge. The C-terminal amino acid has a different environment than the N-terminal amino acid. Yeah?
Could there be side chains that could affect the pKa values?
Do the side chains impact the pKa values of the ammonium at the N-terminus and the carboxylate at the C-terminus? Yes, they can. Not a lot. If you look at the table in your text that shows pKa values for each of the amino acids at least, you see that all the carboxylic acid groups have pKa's in the range of 9. It varies from 9.1 to 9.2, but it doesn't move the needle very much. And that's why it's not worth knowing what the numbers are. Yeah. Okay. Others? Yeah. So when biology synthesizes peptides, biology goes N to C. In the lab, we go C to N for reasons which I would love to tell you about, but we probably won't get to in this class. Aww. But if you want to know more, come talk to folks in my lab. They can tell you more about it. All right, N-terminus, C-terminus, internal amino acid. Now, before we even get to the side chains, there's some things you need to know about peptide and protein structure that will help you understand the crazy, crazy world of alpha helices and beta sheets. So we want to look at this amino acid 2, which is going to be sort of the typical environment for an amino acid within a protein. Any protein is going to have one N-terminal amino acid and one C-terminal amino acid. Most of their amino acids are going to be in this environment of amino acid 2. They're going to have an amide bond on one side and another amide bond on the other side. So we're going to sort of zoom in and look at amino acid 2 and think about all of the bonds in the backbone and what conformations are available to those bonds. So let's look at, sorry, that squiggly line was fine where it was. I don't know why I deleted it. So now we're going to go to something I've already drawn. Are you nervous yet? Make it stop. Okay, fine. What we're going to do is I'm going to copy this tripeptide. I'm going to paste it again, but actually I'm going to say that this is a 3 amino acid segment that's embedded in a larger protein. So I'm just going to put NR here where that's going to be the rest of the protein on that side. And then I'm going to put NH carbonyl oxygen and R there. Okay? I'm going to delete the squiggly lines, and for fun, I got to get the color scheme right here. It looks like it's orange, purple, and green. So we're going to highlight this amino acid as orange. We're going to highlight the next amino acid as purple, and we're going to highlight the last amino acid as green. These are the 3 amino acid segments in our peptide that we're going to focus on. And we're going to think about the bonds in the backbone. And remember, the backbone involves amide nitrogen, alpha carbon, and carbonyl carbon. And we're going to start by thinking about rotation around this bond between the carbonyl carbon of the orange amino acid and the amide nitrogen of the purple amino acid. As I said before, that's an amide bond. What do you know about that nitrogen? It's got lone pairs, but what hybridized is it? sp2, right? And the reason for that is these lone pairs are tied up in resonance with the carbonyl carbon. There's some partial double bond character to that amide bond with a resonance structure that looks like this, right? So that tells you that that amide bond should be shorter than a typical carbon-nitrogen bond. It also tells you that both the carbonyl carbon in orange and the amide nitrogen in purple should be sp2 hybridized trigonal planar. And indeed, for there to be a double bond character between the carbonyl carbon and the amide nitrogen, they need to be, those 2 trigonal planar atoms need to be in the same plane, as though they were part of a double bond. So, if we looked down, sorry, I'm going to add another page. I thought we were going to, oh, that's why. Okay. Scrolling up there. All right, that's fine. We can start here. So we've got the same, I'll just add another page and we'll keep going. Okay, so if we looked down the bond between the carbonyl carbon here in orange and then the amide nitrogen in purple, you can put your Newman projection I there and then think about what you would see. We will talk about the rotation. We're going to describe the rotation about this amide bond with a Greek letter omega. Omega is going to be a dihedral angle that describes rotation around that amide bond. And so we're going to draw a Newman projection looking down that amide bond. We're looking straight at the carbonyl carbon. So the carbonyl oxygen should be pointing up, and then the the alpha carbon here should be sort of pointing down. And we're drawing it this way to reflect, this is what you would see if you looked at a trigonal planar carbon on the edge. So here is the orange alpha carbon, and this is the carbonyl carbon. In the back, where we can't see it, is the amide nitrogen. But if you think about us looking down this bond, we ought to see the NH, the hydrogen here. This is the purple hydrogen. And then on the other side, pointing upward, we ought to see the purple alpha carbon, which I will illustrate like this. Okay, that's thinking way back to 351. I know you're having to access some deeply embedded and perhaps jettisoned information about Newman projections. You sort of see any questions about how I did that? Okay. The angle between the 2 alpha carbons, orange and purple, that angle is going to be omega. Okay? And when omega is 180 degrees, we're going to say that that amide is in the S trans conformation. Some people drop the S and just call it a trans amide. The S refers to the fact that it's technically a sigma bond, but it's got double bond character. If omega were 0, that is, if the 2 alpha carbons lined up in that dihedral angle, we would call that the S-cis configuration. Amides can interchange between the S-trans configuration or conformation and the S-cis conformation, but generally the S-trans conformation is considered to be more stable. And so most amide bonds within proteins are in the S-cis conformation. Rotation from S-trans to S-cis is kind of slow for reasons that we could talk about maybe another time. There are enzymes whose specific job is to help that process go faster. And there are certain amino acids that are okay being in the S-cis conformation. We will point that out in the future. But that's the first amino acid, that's the first dihedral angle you need to know about. That's the omega, and that describes the amide bonds. So you need to think about amide bonds in proteins as being planar. You need to not think that they can rotate and adopt any given value, but that they adopt two specific values, cis or trans, and only those. Okay? Unlike a double bond, isomerization from cis to trans is allowed and can happen by rotation, but it's slow. Okay, any questions about that one? All during sacrament meeting now, you're going to be like, instead of listening to the speaker. Go ahead.
So, is the reason why the nitrogen's on the backbone never gain extra hydrogen charged because they have that, their extra electrons are in the orbital there?
Yeah, is the reason, oh, right. So before, I think you're asking about an exam question, right? On exam 1, you had a lot of groups like this, which you totally ignored. You didn't worry about them picking up protons. Right. The reason that they are not basic is because those lone pairs are stabilized by resonance. They're in a p orbital in resonance with the carbonyl. The pKa for that, what you're describing, you're asking, hey, what's the pKa of this conjugate acid where the amide's protonated? Pretty high. You can protonate it with a really strong acid, and in fact, that's some of what's going on in your stomach that's why you have stomach acid, to help break these amide bonds in your peptides and proteins. You also need enzymes to help the process along too, because amides are very stable. I saw another hand, yeah.
So did you say the peptide bonds where the arrow is pointing, that's in the cis conformation?
Right now, as I've drawn it here, it's S-trans. Because if we look at the Newman projection, the alpha carbon on one side is opposite the alpha carbon on the other side. Yep. Okay. We got 2 more dihedral angles to do, and then we're going to talk amino acids, what the side chains are. So the second dihedral angle to worry about, I'm going to recopy our tripeptide, is now we got to put our Newman projection eyes, and we're going to look down the bond between the amide nitrogen and the carbonyl carbon. And if you want to turn your Newman projection eyes into sort of Emperor Palpatine or Anakin Sith eyes and shout, "I hate you" at me while you're doing this kind of analysis, go for it. Doesn't hurt my feelings. People have written on their exams before, "Bring it on, Dr. Price." It's always fun to see that after the fact. Whatever trash talk you need to do to be successful, you go ahead and feel free to do that. Now, let's look down the bond between the nitrogen and the purple nitrogen and the purple alpha carbon. This particular bond between nitrogen and alpha carbon, the relevant dihedral angle, we're going to give the Greek letter phi. And that's drawn like that. Okay? So phi describes rotation around that bond. If we draw that Newman projection, we're looking straight at the nitrogen. From our perspective, the NH is pointing down, and then the carbonyl carbon from the previous amino acid is pointing up. So to indicate that that's the previous amino acid, I'm labeling it orange. I'll label the nitrogen and the proton purple. Then the carbon in the back, and notice we've drawn this, we're looking at it on its edge, but that nitrogen, as I've said, is sp2 hybridized. Not so for the purple alpha carbon, which is sp3. So that's going to look like, you remember sp3 carbons in a Newman projection look like this. So the alpha carbon in the back is going to have 3 things it's attached to. One of them is the side chain. Another is the alpha proton, which I'm drawing here. And then the third is the purple carbonyl carbon. This dihedral angle can adopt a large variety of values. I'm going to just pick one and draw it, but there's rotation allowed, and it's pretty easy to do about this sigma bond between the amide nitrogen and the alpha carbon. So from our perspective here, it kind of looks like the R side chain should be moving out to our right, the alpha proton should be about out to our left, and then the purple alpha carbon, which I'm going to draw like this. Sorry, I'm just going to use an R group there. Hopefully that doesn't bother you too much. The purple alpha carbon will draw down here. Phi is the angle between the two carbonyl carbons. Okay, that is phi. And phi generally adopts values between, you don't necessarily need to memorize this, but it generally adopts values between negative 180 and 0. Now, a word about the sign of the dihedral angle. That's purely determined by the math. If you can do dot products, you could figure out what it is, but I don't care. And I'm never going to ask you to determine, is that dihedral angle positive or negative? Because nobody does that by hand. We just ask the computer and it tells us. Nevertheless, you can see based on, you can see now here an important place where structure influences conformation and function. Think about the impact of this side chain R, the purple side chain, on that dihedral angle. You can imagine that if the side chain were huge, you might have some steric clashing between the carbonyl and the side chain. That might bias you towards rotating that side chain away, the purple side chain away from the orange carbonyl carbon. This is one way that side chain structure can influence dihedral angles. You will see at some point in the next couple of days that what dihedral angles we adopt, what phi values we adopt, determine whether your peptide is a helix or a sheet or something else. Okay, are there questions about that definition? Okay, this is, by the way, not in your text. Your text doesn't do this. I don't care that your text doesn't do this. I think it's important. Your biochemistry professor will do it, but I like it so much that we're doing it here too. And when you learn it, then it'll make a lot more sense. All right, other questions about that angle? Yeah. If phi were 0, they would be eclipsed with each other. We don't use cis and trans for the phi angle because it's between an sp2 carbon and an sp3 carbon, and so you'd have to talk about, you'd have to define what you were talking about. But no, yes, you could say that if phi were 0, the carbonyls would be eclipsed. Others? Okay, let's do the last dihedral angle, which is going to be the next bond over. That is the bond between the alpha carbon and the carbonyl carbon. So I'm going to copy our peptide one more time. And this time we're going to put our evil Sith eyes right behind the alpha carbon. Why are Sith jaundiced? I don't know. Bloodshot and jaundiced. That doesn't necessarily argue that that's how you want to live your life, right? That being said, I did dress up in my Darth Vader costume last night at Special Needs Mutual, and it was epic. It was our Halloween party. By the way, it's so, it seems like a good idea to be in a Darth Vader costume, but you can't move, you can't see, and it's really, really hot inside. It was very unpleasant, but I looked awesome. And since appearance is everything, right? Okay, so let's look down. Please do not write me an email that appearance isn't everything. That was said in satire. All right, the alpha carbon's in the front. We're looking down the bond between the alpha carbon and the subsequent carbonyl carbon. From our perspective, the R side chain would be coming out to our right. The hydrogen would be out to our left. And then the amide nitrogen, the purple amide nitrogen would sort of be pointing down. And I'm just going to abbreviate it like that with an R group. These are all the purple groups. In the back, we're going to see pointing down sort of towards the bottom is the carbonyl oxygen, still the purple carbonyl oxygen. And then on the other side, we're going to see the green amide nitrogen. The dihedral angle here is the angle between the green nitrogen in the back and the purple nitrogen in the front. We give that dihedral letter angle the Greek letter psi, which, if you're a Teenage Mutant Ninja Turtle fan, looks like the sword, which is also called a psi, that Raphael used to carry around. That was a thing in the 80s. I don't know whether it's been even noticed by your generation, but so be it. Okay, that dihedral angle can adopt values that are a little bit more broad. It can go from 180 all the way through to 0 and all the way through to minus 180 on the other side. Quite a bit more possibilities, but you can again see how the identity of that side chain might influence whether it's better to have the green and the purple nitrogens to be further apart, or whether you want the green nitrogen in the back to be far from the side chain. So you've got three dihedral angles, omega, phi, and psi. Omega can only have two values, cis or trans, and then phi and psi can have a lot more values. Now, why are we talking about this? And the reason is because the values that are adopted by these dihedral angles, phi and psi, determine the overall shape of the protein or its conformation. Have you heard of the alpha helix before? Yes-ish, maybe. So if you've ever heard of the alpha helix, somebody's probably drawn it for you, and it's this lovely spiral. The only reason the helix can form a spiral is because the dihedral angles of each amino acid in that helix adopt values that allow the individual amino acid to twist around and upward. So the backbone has a spiral trajectory. I'm going to try to illustrate that now with some pre-drawn figures, which you will hate, but which are going to make a point. You're going to want to know what should you memorize, what should you not. I'll try to give you the take-home message. But you should be curious as to how the structures, how the conformations about these bonds work together synergistically to make a shape like a helix. So, behold, a helix. That's actually a helical trimer. You have one helix here, another helix here, and another helix here. What I'm showing you is a ribbon diagram of an X-ray crystal structure. You'll have to learn about that later. X-ray crystallography is a way that we can assess the three-dimensional structure of a peptide or a protein. Each of the sticks here represents a bond between a carbon, say, and a nitrogen. I've eliminated as much detail as I can, so what we're just doing is we're noticing this sort of ribbon cartoon that traces the backbone of the peptide. If you're interested, you can see side chains branching off at various points. Those are the R groups. We'll talk more about them later. We're going to focus in on this 3 amino acid segment, orange, purple, and green. You like the color consistency? And we're going to look at the phi and psi dihedral angles within this amino acid so that you can see what they have to be in each amino acid so that you can form a helix. What we've done here is simply redefining omega, phi, and psi. We're going to skip omega because it's boring. No, fine. We like to review things we understand. So here we're looking down the bond between orange carbonyl carbon and nitrogen. This is zoomed in on a structure from this helix. In this structure, red atoms are carbon, blue atoms are nitrogen, the colored atoms, orange and green and purple, are carbons. Hydrogens, if shown, will be white. Got it? Okay. So looking down this bond, you can see, if we look at it from the side, you can see what we see in this Newman projection, which is the alpha carbon on the orange amino acid is opposite the alpha carbon on the purple amino acid, or in other words, omega equals minus 180. Okay, do you need another minute to absorb that, or are you following along with sort of the stick diagram? Okay, so now let's move ahead. So that's actually in this helix, the amide bond is a trans amide bond. Yeah?
Do you have drawings of the other two?
I do.
How
Nice segue, thank you. All right, so phi is the next amino acid. We're putting our sithize. I'm glad you asked. Do you want to actually come up here and ask those kinds of questions along?
Yeah, sure.
Okay.
Give me a cue.
You can be like the guy, I forget what his name is. Jimmy Fallon's got the guy that stands over at the podium and makes wisecracks. Color commentary is maybe what we're talking about. All right. By the way, I don't know anything about sports, but I'm convinced I could be a good color commentary. Well, you know, Greg, what you really want to do is go out there and score more points than the other team. Boy, if they get another turnover, that's going to be, yeah. I'm sure there's more to it than that. Please do not write me an email about how disrespectful I have just been about color commentary. Okay. Looking down now the bond, we're going to look for the purple amino acid and see what the phi angle is. We're looking at the bond between nitrogen and alpha carbon. Here is that view. You can see we've got, in this particular alpha helix, the phi angle is between the carbonyl carbon on the orange in the front and the purple carbonyl carbon in the back. And you can see that that dihedral angle is small. In fact, the side chain, the purple side chain, is eclipsed with the amide proton on the front carbon. This dihedral angle has a value, it's around -60. And it needs to be that way in order for the amino acid to tilt the next amino acid upward in a spiral. If that number is not -60, you don't got a helix. Okay, so that's the phi angle. I've drawn the Newman projection here, and if you want to download these notes and look at it and go through it a little bit more slowly, you can convince yourself that I've drawn it correctly. The next dihedral angle is psi. We're going to look down. Oh, where did it go? Oh, I inserted a page at the inopportune moment. Here we are. Here's the same amino acid zoomed in this structure. Here is the alpha carbon and the carbonyl carbon. We're looking down this bond, and here is that Newman projection view. Here I've highlighted where the amide nitrogen is, the purple amide nitrogen in the front, the green amide nitrogen in the back. You can see again that that dihedral angle is small. You can see again that the purple side chain isn't really eclipsed with anything. It's sort of bisecting the space between the carbonyl oxygen in the back and the amide nitrogen in the back as well. So this dihedral angle in an alpha helix is around -50. Has to be, otherwise you don't get the right tilt of the amino acid setting up the next amino acid. If you have a segment of 4 or 5 amino acids that all have this dihedral angle, you now have a helix. It takes 4 amino acids with these dihedral angles, or rather 3.5, to get 1 turn of a helix. If you put 7 of them in a row, you get 2 turns of a helix. 14, you get 4, and so on. And so alpha helical secondary structure really comes from these dihedral angles, adopting a specific value. Now, if we could talk about beta sheets and what dihedral angles are typical for beta sheets. We won't, but that's an orientation into peptide structure. Now, we need to talk, any questions you want to ask about that? Yeah.
You said omega is always between 0 and -180.
Omega is always either 0 or 180. Phi, yeah. Yeah.
So does that mean they always rotate in the same direction?
Yes, that's a fantastic question. Thank you for asking that. Do all helices rotate in the same direction? Yes. The reason for that is that it's the side chain that determines if you tried to do it the other way, suppose you tried to go the opposite direction, instead of -60 plus 60, instead of -50 for psi plus 50, you would form a helix in the other that would twist the other way. But if you do that, the side chain starts bumping into stuff. The chirality, this is beautiful, the chirality of the side chain determines the twist of the helix. Yeah, so again, there's more we could talk about there. I would love to, but that's probably deeper than the scope of what we want to do. One other thing I want to say about phi-psi angles is that you can plot them together on a 2D plot of phi angles versus psi angles. You can measure those angles for each amino acid in a structure, and the plot will tell you something about the structure. The plot's named after the chemist that originally envisioned it, Ramachandran, and you plot phi on this axis, the x-axis, and psi on the other one. And you see for a protein that's alpha helical, all the phi psi values cluster in this area around -60 on this axis, the phi axis, and around -40 or 50 on the psi axis. If you have a protein and you plot all the phi psi angles and they're all here, you don't even need to see the structure. You're pretty sure it's an alpha helix. There are other values that are allowed if they were up here. This combination in this region of the Ramachandran plot would be more consistent with beta sheet structure. And I'll show you some more examples of that. However, I want to talk now about how the side chain affects the preferred structures of an amino acid. Any other questions you want to ask about that? If you want to see some of these steric clashes, there's a link in the study guide to a little web page where you can actually dial in what the phi psi values are. It'll show you in big red labeling anytime your phi psi angle makes steric clashes occur. And it's kind of interesting. It gives you a sense for why things have to be the way they are. Go ahead.
So for helices, we talked about normal angles for phi and psi.
Yeah.
But omega can be either 0 or 180.
Yeah.
And for helix, what's it?
In a helix, omega is always 180, always trans. Yep. Okay, good. Here is an example of a Ramachandran plot that's not specific for alpha helices, but it's for like all proteins. And so you can see most of the phi angles in all proteins are less than 0, but the psi angles vary quite broadly. There is a weird segment here that shows up with dihedral angles that would be more consistent with an alpha helix, only they're opposite. Don't worry about that. There's more to say there, and I would really love to, but it's not going to be worth the squeeze. However, the juice won't be worth the squeeze. However, there are some amino acids you will see that are better at adopting these kinds of dihedral angles. I'll give you a hint. One of them that's really good at it doesn't have a side chain at all. That's glycine. And it's because it doesn't have the steric clashes that the other amino acids do when they adopt these kinds of angles. Okay, so in the last 5 minutes, we are going to memorize all 20 amino acid side chains. Yeah, and their pKa's. And we're going to, this is going to take some time on your part, and you should do whatever it takes to memorize stuff. Normally in this class, we have not been heavy on memorizing things. We've not been serious on information recall. However, memorizing the amino acid side chain structure is absolutely key to understanding the connection between sequence, structure, and function in proteins. That's an important topic. It's a Nobel Prize winner this year. It's something that I feel like 20 years from now, when you tell somebody you had a class from me at BYU, I want you to be able to say that I told you this, even if you didn't care about it, okay? This is like when I'm brought before the judgment seat of God, I want to be able to say that I told you guys this. I am discharging my sacred obligation. So you're going to like it, is basically what we're down to. Okay, I have drawn a diagram, yeah, right? Yeah, isn't it beautiful? So we're going to go through each one. I've clustered them in groups of similarity, and we're going to talk about each one. You're going to need to know the name, the one-letter code, and the side chain structure. You're also going to need to know, where applicable, the side chain pKa. Now, on this diagram, I have given you a mini Ramachandran plot that shows you for all proteins a heat map of what dihedral angles are generally preferred by that amino acid. That's bonus. You can ignore that, but it's actually really cool. Because, for example, alanine, which just has a methyl group on the side chain, here is the alpha carbon. I'm not showing anything else. The backbone's exactly the same. Alanine with just a methyl group has a lot of cases where it's in an alpha helical conformation. And it turns out alanine's quite comfortable in an alpha helix. The side chain's small, there's not a lot of steric clashes, though it's also pretty comfortable in beta sheets. So alanine you will find in lots of contexts. Not so for some of the other amino acids. The green group on our drawing here is of the relatively non-polar amino acids. These are generally amino acids without heteroatoms in the backbone. Some chemists will talk about carbons in amino acids with Greek letters. So notice for alanine, the beta carbon is a methyl group. That's the one that's adjacent to the alpha carbon. You don't need to memorize the Greek letters, but I'm going to use them in class when talking about things, so just sort of be able to know what I'm talking about. Valine is nice because its one-letter code is V, and the side chain sort of looks like a V on the beta carbon. Valine is an example of what I call a beta-branched amino acid. In other words, on the beta carbon, you got two other substituents instead of just one. Notice what that beta branching does to valine. This is not important, but you can see alanine had two unique environments up here, two different types of dihedral angles it was comfortable with adopting in this region of the plot. You put that second beta branch, you put that beta branch group on there, the second methyl group, it knocks one of them out. In fact, valine is more comfortable. There are more examples of valine in a beta sheet than there are of alanine. So now you begin to see how structure and sequence is related to conformation and ultimately function. If you wanted a peptide to be in a beta sheet, valine would be a decent choice. I want to talk about all of these because I don't have a favorite one. They are all wonderful. But I am also reluctant to spend a ton of time talking about things that you're going to wonder, well, do I need to know that or not? I've tried to make it absolutely clear. You better be able to draw the correct structure of leucine. If I write a one-letter code, you better be able to know what I'm talking about with a particular amino acid. So these are the nonpolar ones: alanine, valine, leucine, isoleucine, and methionine. Methionine is interesting because it is the first amino acid, and that's a start codon in ribosomal peptide synthesis. It's the first amino acid in most peptides. A lot of eukaryotic proteins, that methionine gets processed off. Methionine is considered to be nonpolar, though it has a sulfur heteroatom. It's got, between the alpha carbon and the sulfur, you got two carbons, and then a sulfur, and then a methyl group. Pretty flexible. It's easily oxidizable, which doesn't matter, but if you come work in my lab, it does matter. This modification of the sulfur to this sulfoxide is spontaneous. It happens in air, and it's pretty annoying. Some people will group glycine in with these nonpolar amino acids because glycine doesn't have a side chain, it just has a CH. I don't, because glycine is weird. If we zoom in and look at glycine, I want you to compare the Ramachandran plot for alanine. See it? You're taking a mental picture. It's memorized. Good. And now do oh, no, that's not what we wanted to have happen. Now let's zoom in on glycine. How is that different? Alanine was leaning to the left, right? It was biased to the left. Glycine, which doesn't have the stereocenter, is equally as comfortable here in this side as it is over here. That's because it lacks the steric side chain. Yeah. Should you know which one's polar, nonpolar? Yes, but if you can look at the structure, if you memorize the structure, that answer should come automatically. Right, so, and I'll answer this question more next time, but if I say leucine and you've memorized the structure, you should be able to tell from the structure whether it's polar or not. All right, have a good weekend. See you next time.